a very innovative combination of electrospray, ion mobility, and the QTOF mass analyzer. And I believe we're going to hear an application of that technology this morning from Ben Collins. Ben is from the Queen's University of Belfast, and he speaks on behalf of Bruker. Before turning it over to Ben, I'll just remind everyone, please use the Q&A function to ask questions. There will be some time at the end to address those. So with that, I'll turn it over to Ben. Welcome.
Thanks, David. I'm just having a little issue trying to get the right screen shared here. So are we seeing the full slide view or the presenter view right now?
It's good, Ben.
It's good. Okay, I'm sorry. Sorry, I thought I had this on the wrong screen there. Okay, sorry about that. I want to, great. So I want to thank US HUPO and Bruker for the chance to present here today at this virtual conference. So I'm going to be talking about DIA-PASEF. So let's go. So here's a quick outline of what I want to talk about. I'm going to start with some background on the idea of trapped ion mobility and PASEF for those who are not familiar with that. I'd like to talk about the concept of DIA-PASEF and what the potential benefits might be there. Then I'm going to want to show some data that gives an idea of the performance of the method. And lastly, I'll try and show a kind of first glance of a biology data set and one interesting finding in the area of chronic lymphoid leukemia. So let's start with TIMS and PASEF. And so here we see this nice overview of the various kind of flavors of ion mobility separations that are currently in routine use. This I stole from a very nice review paper from Aaron Baker's lab, which I think is actually very helpful for those who might be new and getting into ion mobility. And so starting from the left to the right, we have drift tube separations, which is probably the kind of simplest to understand, and the variety maybe most people are familiar with, where ions are being propelled through an inert gas, an inert buffered gas using an electric field, and essentially the force imparted on each ion is going to be the same for a given charge state, but the resistance that each ion encounters is going to depend on its collisional cross section. So the smaller ions are going to get through the drift tube first. Up next then will be traveling wave, which is similar, except that the idea is that the ions are kind of being bumped along by a traveling wave voltage, which is moving through the cell. And then in the middle is TIMS. So this is the one, trapped ion mobility, which I'm going to be going into more detail on the next slides. And then on the right, you have field asymmetric or differential mobility separations. And I think these can be thought of as essentially more like filtering methods. And I think it's worth pointing out now that we're in the very nice position that there are commercial implementations of all of these strategies, and they're really becoming, it seems, more and more popular as a way to achieve a kind of an additional level of separation in combination with mass spectrometry. But yeah, so the one I want to talk about primarily is trapped ion mobility. So this is a flavor of ion mobility separations that was recently developed by Bruker and now implemented commercially on this timsTOF Pro platform. And I think you can, for those who aren't familiar, I think you can kind of think of TIMS as somewhat the reverse of classical drift tube ion mobility separations, where what we have is a carrier gas which is moving through this concentric ring electrode tunnel toward the mass spectrometer, and this is being opposed by a ramped electrical field, which is weaker at the front of the cell and stronger towards the back. And so the force of this carrier gas on an ion is going to be determined by the collisional cross-section of that ion. So this means that larger ions are being pushed deeper into the mobility cell, and essentially all ions are going to migrate to the point where the force imparted by this carrier gas is balanced out by the opposing electrical field. And this is how the separation is achieved. And so what we have is a mixed population of ions entering the cell, which is being separated and trapped, essentially can be trapped indefinitely. But the clever bit here is that this voltage ramp, which is holding back the ions, can be sequentially dropped, such that the ions are now going to enter the mass spectrometer serially, right? So TIMS is separating the ions, but it's also focusing them into packets of something on the order of 5 milliseconds from an accumulation time of something like 100 milliseconds. And there's quite a substantial focusing of the ions from a flux, from this flux into more discrete packets. And I think that people tend to focus on TIMS from the perspective of separating power, but this kind of focusing effect is also arguably as important or possibly more important. And then at some point, Brooker realized that if they were to use two TIMS traps in series, they could get the efficiency to go up. So ions which are being accumulated in the first trap for 100 milliseconds and then move to the second trap where they're scanned out for 100 milliseconds. And then concurrently, you have a new set of ions being accumulated in the first trap. And this allows you to, this sort of parallelization allows the efficiency to be kept something pretty close to 100%. And so Brooker have commercialized this, and as I mentioned, in the form of this timsTOF Pro instrument. And so this is essentially a conventional QTOF mass spectrometer with this dual TIMS mobility cell bolted onto the front. And I just show this schematic just to remind people that the ion mobility separations are happening on this instrument in front of the mass spectrometer, right? So that all the mobility separations are happening at the precursor level before we ever get to any precursor selection in the quadrupole or any fragmentation happening in the collision cell. And so a few years back, Matthias Mann's lab partnered with Brooker to develop a DDA implementation that leveraged this idea. So this ion concentration, ion focusing effect, in addition to the fact that the ions were being introduced serially into the mass spectrometer, this allowed them to do very fast and very sensitive DDA analysis. And they call this parallel accumulation, serial fragmentation, or PASSIF. And I cite a couple of papers below from Florian Meyer if you're interested in learning more about this. So this is the background on TIMS and on PASSIF. So let's move on and talk about how we can exploit these characteristics in DIA mode. So Rudy already mentioned a little bit that, well, around 2 years ago, we had been following this work from the Mann lab and from Brooker, and we started to wonder whether this could be a potentially useful idea in DIA mode. And it turns out actually they were wondering very much the same thing, and so we were happy to kind of team up in this and try and do it together. So this project is led by Florian Meyer in the Mann lab, pictured top left there, and then Hannes Rust and his group at the University of Toronto are developing the data analysis pipeline. And I started this when I was still in Zurich with Rudy, and now we're continuing to work on this in my new lab in Belfast. And Brooker have done, of course, all the work to implement all these new scan modes on the timsTOF Pro instrument. Okay, so if you're interested in more detail of what I'm going to describe now, I just want to mention we uploaded a sort of an early preprint on this around ASMS time last year with some rather early data, but we just actually incremented this preprint version a few weeks ago, or maybe 1 or 2 months ago by now, to a somewhat more mature version, and contains some more of the data that I'm going to be showing today. And also, actually, it includes, if people are interested to take a look at the raw data, they can find details to access this in the second version of the bioRxiv preprint. Okay, so what are the problems that we might actually be able to reasonably address using this kind of combination of DIA and PASSIF? And I think the most interesting one, I think, is this sort of global efficiency of ion utilization at the mass selective quadrupole. So typically the efficiency of ion utilization at the quadrupole in DIA or swath mode is something like 1 divided by the number of precursor isolation windows. So something like 1 divided by 32 or 1 divided by 64. So it's something in the range of maybe 1 to 3%, something like this. And of course, for DDA mode, this is going to be actually much lower than that. Yeah, so obviously all ions outside of this whatever the current quadrupole position are being lost. And the question is, can we improve this by synchronizing the quadrupole with the ion mobility separations in DIA mode? The second point is we know that selectivity can be sometimes limiting in DIA mode. We're often kind of operating close to the selectivity limits there. And mobility separation, it seems, could help to reduce interferences. And this has been showed to some extent before in other methods, like for example, HDMSE. And we were interested to see how much this would help. And third, I think we can just also benefit from this ion focusing effect implemented in the PASEF approach. And so we also just wanted to have this benefit in DIA mode. Okay, so now I want to explain what this method actually looks like schematically. So here we can see a kind of a typical picture of what the ion mobility resolved mass spectra look like for MS1 scans. And this is what one cycle of the 100 millisecond cycle of the ion mobility cell looks like, but now it's being averaged actually for all retention times across a single injection of HeLa. And I think the way that you want to think about this picture is that time is sort of running from the top to the bottom of the graph, right? So in 100 millisecond cycle of the TIMS device, there's going to be something like 1,000 TOF pushes, and what we're looking at are those spectra kind of stacked on top of each other from top to bottom in this heat map. And each of these little features you can see represents a peptide precursor. And I think what you can appreciate is that the ion mobility and the mass to charge are pretty well correlated. But the charge states are somewhat separated out, with the singly charged species being pretty well separated from the multiply charged species. And so now if we go into DIA mode, where we're talking about MS2 spectra, what we can do is we can use the correlation of this correlation of the MZ and ion mobility to position the quadrupole where we know there should be bigger ions at the start of the mobility scan, right? So if you remember, I said that the way this is working is that the bigger ions are being released into the instrument first. And so as this ion mobility scan proceeds, we can shift this quadrupole position over from larger to smaller MZ. And each of these kind of blue lines in the animation here represents kind of one of 1,000 ion mobility resolved MS2 scans with one of several different quadrupole positions. And the idea here is that by using the correlation between MZ and ion mobility, we should be able to basically increase the global ion utilization efficiency by at the mass selective quadrupole, right? We sort of trying to move the quadrupole to catch ions coming out of the mobility cell. And this is basically, I would say, this is the basic concept that we started with, that we had towards the start of the project. Now, it turns out that this scheme that I described in the previous slide is maybe not completely ideal for complex samples, because the isolation windows are too big in the mass dimension, and you run into selectivity issues. And so we need to probably make some sacrifices in terms of the ion utilization efficiency in order to get the selectivity that we want. And so this means reducing the window size in the mass dimension. And what this means is that we need to probably spread out these isolation windows across multiple cycles of the trapped ion mobility device. And so this should allow us to get the selectivity to go up a little bit, even at the cost of the ion efficiency going down a little bit. So what's shown in this picture are kind of two examples of isolation schemes that try to illustrate this. And in these pictures, the boxes show the isolation windows, where the ones that are filled in solid white are the first cycle of the TIMS, and then these numbers are indicating the kind of subsequent cycles. So the lower left one, we call this kind of standard DIA PASEF, and this is, we would say, I guess, a sort of general purpose method using 25 M/Z windows, but actually takes, which you can see are kind of stacked on top of each other to cover the whole precursor space. And so this means it takes 16 cycles of the TIMS device to cover all of this precursor space. And then the upper one, which we call sort of high sensitivity, is sacrificing a little bit of selectivity by using a bit bigger windows in the mass domain. So these are 50 MZ wide. So there's going to be potentially a little bit more interferences, and it also may have a little bit lower precursor coverage than the standard method. But the benefit is that it's only using 4 cycles of the ion mobility device. So our expectation is this is going to be more efficient. And then you can see in this graph on the right side there that the raw signal actually increases substantially beyond standard DDA or DIA. So DDA, this is essentially the MS2 ion current, and in DDA is in red. DIA, a standard DIA method, is in gray, and then the sort of standard DIA PASEF and then the high sensitivity DIA PASEF are in green and blue. So it seems like we are indeed getting more ions depending on which scan mode we're using. So that's basically the concept. The next question is what do we need to do differently in terms of the data analysis? So we had to make some adaptations to this OpenSWAT software to analyze the data. And so probably know in conventional sort of DIA or swath type approaches, we're acquiring MS2 spectra from wide precursor isolation windows deterministically. And normally the data analysis would be to, a pipeline would want to extract ion chromatograms for fragment ions in this series. But in DIA-PASEF, we have each of these MS2 spectra from each push of the TOF is being stored in a mobility resolved way. So if we know in advance something about what the ion mobility of a particular target peptide is, like for example from a library, then we can selectively extract just the scans at that value with some tolerance. And this is, so this is the main adaptation that had to be made to the software. This was in addition to some changes to the scoring, but this is the main part here. And so because the MS2 scans are ion mobilities resolved, we can just selectively look at the scans where we know the signal should be. And the other thing I want to say on the software side is that this currently the data analysis pipeline is library based. Of course, you could imagine other ways to do this potentially, but at the moment we're using libraries. So essentially what we're going to be doing is acquiring DDA PASEF data on representative samples or usually offline separated fractions. We're going to search this data with MaxQuant, and then we're going to transform the results of the MaxQuant search into an OpenSWAT style library using some adapters. And essentially this is going to look like a standard OpenSWAT library, with the exception that there's one extra column or one extra data field which is containing the ion mobility for the target peptide. Okay, so let's get into some results now. So here's some representative results. In fact, Rudy showed already a version of this slide from 200 nanograms of HeLa samples analyzed with a 2-hour gradient using this standard DIA-PASEF method, which I just showed the little picture again on the right side to remind you. And we detect something in the range of 65,000 to 70,000 peptide precursors per run, which are mapping to around 7,500 proteins. And currently we're only using proteotypic peptides in a low redundancy database for protein inference, so potentially this could be a bit higher if we were to implement protein grouping. So this and the set of things that we detect represent about 85% coverage of the library that we have. And I should say that this library has been generated from the same sample using 24 basic reverse phase fractions analyzed in DDA-PASEF mode, which results in a library of about 9,500 proteins. And so we're getting about 85% of this back in the single shot DIA-PASEF analysis. And if we map this onto estimated copy numbers per cell for these HeLa cells, we cover something like 4+ orders of magnitude at the protein level. And we can also see that the data completeness here across technical replicates seems to be pretty good, which is sort of 99% of proteins are complete out to around 7,000 proteins. So of course, being able to detect peptides and proteins is one thing, but I guess what actually really counts and what most people want to do is quantification. So we decided to perform one of these LFQ bench style mixture proteome experiments where we took 200 nanograms of HeLa digest and spiked this into, we spiked in either 15 nanograms or 45 nanograms of yeast digest. So the human ratio should be basically 1:1, and the yeast ratio should be 3:1. And this is essentially what we see in these kind of blue and green clouds respectively, which are more or less centered on the expected ratios. And shown in this lower right corner, you can see the numbers of inferred proteins that we can detect, and also the ones that we can get a ratio for. So here we have about 6,700 HeLa proteins quantified and around 700 yeast proteins. And I guess I would say that the results are pretty comparable quantitatively to other platforms. I think one difference here is that we're probably injecting quite a bit less material, and we seem to maintain pretty good quantification even when constrained by this lower amount, 15 nanograms of yeast, which is really not much material. And we can also ask the question whether this selective extraction in the ion mobility space actually helps to clean up the signals. So here's an example of a peptide where we can see the MS2 fragment ion chromatograms And in the upper panel, this is the signal essentially if we don't restrict the signal extraction in the ion mobility space in the open swath analysis. And the lower one is if we do restrict the extraction in the ion mobility space. And you can see that this seems to make quite a difference. And in this panel on the right, this is what the trend looks like globally. So for all the peptides detected, if we look at the log signal to noise ratio, either when we're not applying this restricted extraction on the left, or else when we're applying a progressively narrower extraction window going to the right, that this has a substantial impact on the signal to noise. Then we can also ask, does the selectivity improvement make a difference in the boost the detection numbers? And this seems to be the case at the peptide precursor level, which goes up around 25%, but also at the protein level. So it seems like this is certainly worth doing. Okay, so in the data I've shown until now, we've been injecting something around 200 nanograms, and this is already not so much starting material, I would say, but we were interested to see what would happen if we push this down lower into the nanogram range. So here I'm going to come back to these two different acquisition protocols, which I mentioned earlier on, which were the so-called kind of standard method, which the previous data came from, and also this higher sensitivity method. And just to remind you, the difference being here that this high sensitivity method is sacrificing some selectivity with wider mass precursor isolation windows in order to get higher efficiency in terms of ion usage. And so if we compare these two methods on the lower left there, just analyzing HeLa samples, we see that we get, as expected, higher intensities for the higher sensitivity method. And then interestingly, so this is plotted against the coefficient of variation, and you can see that interestingly, this results in a little bit better precision. If we look at the apparent dynamic range, we can also see that we seem to be observing a little bit more extra dynamic range using this higher sensitivity method. But if we look at the number of proteins detected, we can see something interesting here. So it seems like there's sort of a crossover between the methods depending on how much material you load. So if we look on the right-hand side where we're loading 100 nanograms, the standard method seems to do better in terms of the number of detected proteins, but as we go down towards 10 nanograms, the high sensitivity method seems to kind of start to creep ahead in terms of the number of proteins detected. And this I think is interesting. It's probably indicating that when we have a lot of material, we're more selectivity limited, but as you get down to these low amounts of material, we're potentially somewhat more sensitivity limited. And so to me, this is pretty interesting, and says kind of indicates to me that there's could be more room for improvement and optimization. You know, you could optimize the isolation schemes for given types of projects, because there's a sort of a largish parameter space to be further explored here. Okay, so in this last section, I just want to briefly show one or two pieces of data from a more kind of biology-oriented study. In this case, we're looking at chronic lymphoid leukemia. So this is the most common leukemia in Western nations, and the clinicians are particularly interested in this trisomy 12 in chronic lymphoid leukemia. So this is occurring in 16% of all cases. It's an indicator for poor prognosis, and potentially also has implications for altered drug response. And the question here is, what's the effect of this trisomy 12 on proteins and maybe eventually protein complexes in CLL or potentially signaling pathways? And I should say this is work led by Fabienne Meyer-Abd at the University of Zurich Hospital, she's a clinical hematologist there. And this kind of complex looking graph is just to show you the set of patients that we're looking at. So this is 50 CLL patients, and on the top part is indicated all the different kinds of genetic lesions they can have, that the patients have. And I just want to mention the top one circled in red. This is this trisomy 12 that we're particularly interested in. So about half of these patients have an extra copy of chromosome 12. Incidentally, indicated in blue in this lower part is the other types of kind of molecular data that are available for these samples. So there's quite a large background of data there that we will be able to integrate with. And so we did essentially what I've described was to analyze these 50 patients, 200 nanograms of material from these 50 patients in DIA PASEF mode, also some of them in DDA PASEF mode to generate a library, not with fractionation in this case, so this might be, the library may be somewhat limiting in this case, but we may go back and look at that. We search the DDA data with MaxQuant, and we do the analysis with OpenSWAT, as I mentioned. And so this is the number of inferred proteins we can detect from these samples. It's something a little bit more than 6,000 proteins from around 55,000 peptide precursors, and the data completeness is on the order of 82%. But now, of course, we're talking about biological variation, so this we think is still pretty good. And here we're looking at a hierarchical clustering of the data on the left-hand side and a correlation matrix on the right-hand side. So this is fairly complicated, but this is just to point out there's no particular clustering by genomic lesion or batch effects that we're observing. So these are pretty heterogeneous samples. However, if we kind of zoom in and look at the log ratio, so if we look at the protein changes sorted by chromosome, then the story looks a little bit different. So this plot shows the log fold change for the proteins separated out by chromosome for the trisomy 12 patients divided by the cytogenetically normal patients. And I think what you can notice is that while all these ratios are centered around 0 for all chromosomes, with the exception of chromosome 12, which shows actually a substantial increase in the trisomy 12 patients. And so this increase is not necessarily as much as you would have predicted from the genomics, so it's not 1.5 times more, but we think that there's probably good biological reasons for this, and it seems that the cell probably has mechanisms to deal with these extra proteins, right? So previous data from our lab and then from other labs kind of indicating that in particular protein complexes are important here, and where proteins in complex are probably protected from degradation, proteins kind of floating around in the cell might be more prone to degradation, so the cell is potentially taking those out. And so we're going to be following this up, and Fabienne and her collaborators are currently following up on this data set. Okay, so that brings me already to the summary. So we think that this DIA PASEF method is increasing ion utilization by synchronizing the quadrupole precursor isolation with ion mobility. So this is, I think, the main message. But it's also leveraging this ion focusing effect, which we think has benefits for sensitivity, and also this sort of semi-orthogonal ion mobility separations, which is helping us with selectivity. And at the same time, we have all the usual kind of benefits of DIA. So the deterministic duty cycle that is leading to high quality, MS2 level quantification. We have comprehensive coverage of the precursor space with MS2 spectra as normal. And we think there's pretty promising results from these benchmark samples for identification and quantification, and now also for some more biologically oriented studies like this CLL studies. And this especially seems to be true for lower amounts of samples, and we're looking at always amounts of samples in the range of 10 to 200 nanograms. Okay, so finally, just to acknowledge a few people, I guess I mentioned already the main people on the DIA PASEF development side in the Man Lab, in Hannes Rust's lab in Zurich while I was still there with Rudy, and at Bruker. The timsTOF work at ETH has been helped by Peng Zhu, Claudia Martelli, and Ludovic Gillet. And the CLL project is led by, as I mentioned, Fabienne Meyer-Abt at the University of Zurich Hospital, along with a large team of people there and elsewhere. So with that, I'll finish up. I'll thank you for your attention, and if there are any questions, I'll do my best to answer them.
Okay, great. Thank you for that. Great talk. There are a number of questions here, and I'll moderate those. I'll try to combine some of them that are kind of similar. A number of people have questions about the starting amounts of protein, and your observation that sensitivity seems to be maximized for the lower amounts of protein input. How does that figure into your choices of nano versus micro flow chromatography?
Yeah.
Yeah, go ahead.
Okay, all right. So Yeah, so so I should say, I mean, the reason why we're typically not going too far above 200 nanograms is that at some point the ion mobility cell gets you start to run into saturation effects. So we're usually trying to keep it not too much above this. Yeah, we have tried to use this instrument also with EvoSep, so that's not quite microflow, or maybe it's low microflow. So probably what's happening there is, you know, there's a little bit less signal due to the higher flow rate, so we can probably load a little bit more, but then we're going for shorter gradients, so everything gets compressed, right? So the amount you can load ends up being somewhere similar. So I think for EvoSep, we're loading something in the range of like 100 to 150 nanograms.
Okay. And the related question would be, as you go from the you know, lower to the higher amounts of input, how does that play out in terms of number of peptides versus proteins?
So I guess they are talking about this here. Yeah, I mean, I'm not sure if I quite got the question, but the peptide number.
I think the question comes down to protein inference, and you know, if more peptides or less has an impact there.
Yeah. So okay. So I think I guess it relates to this comparison on this slide here. I think that while we're showing more proteins for 10 nanograms for this kind of higher sensitivity method, it does have less peptides detected than the standard method. So that's certainly the case, if I got it right. Yeah.
Thank you. So a question comes up on sort of the practicalities of you know, instrument maintenance. You know, how often do you need to clean the ion mobility source? Is that, you know, more often than the TOF or Q1? And how does that affect the experiment?
Yeah, so the maintenance is, I mean, the cleaning is surprisingly little. And I don't know why that is. So it could be either to do with the fact that we are loading less material, right? We're only ever loading a couple of 100 nanograms, or it could be to do with the fact that maybe the mobility cell is kind of protecting the rest of the instrument to some extent. But the only routine cleaning that we do is of this glass transfer capillary. So in between the spray and the mobility cell is a dielectric glass capillary. And this we clean around every, I don't know, 2 months or so, 2-3 months, which does require a vent. But we don't clean anything else. And my understanding is beyond that. And my understanding is that the people who have had the instrument for a little bit longer also are not really cleaning beyond this dielectric capillary. So from a robustness perspective, my experience has been pretty good.
Great. So a question here is on the separation of the charge states. The question notes that the 1+ charge states are well resolved, but the 2 and 3+ tend to come together. And what about the higher charge states? What's factoring into these differences?
I don't know if that's completely understood. So I think we would need so yeah, it's certainly the case that the +2 and the +3 are somewhat mixed. Interestingly, the +3 has a kind of a bimodal distribution where one part of it seems to overlap with the +2 and another part of it seems to be slightly away from the +2. And I don't know if that's quite understood why that is. But what you would see for higher charge states is that it would be going basically down to the lower right of this diagram, right? So things are going to moving into the lower right corner. And I think it certainly will be interesting to check if different kinds of peptides that have different charge states distributions or different properties are gonna look different in this separation. So for example, actually I just saw in the last few days or last week or so a preprint on looking at cross-link peptides here, and these are also, so they could show that the cross-link peptides are kind of at least partially separated from monolink peptides, and they were able to target those specifically. So yeah, I think there's a lot of work to do to look at where different kinds of peptides that have different properties are gonna map onto here, and whether that can be leveraged, and whether you can change the parameters of the ion mobility scan in order to improve things.
Okay, so I think maybe you just addressed this one, but there was a question on cross-linked peptides and whether the mobility separation would be useful for those.
Yeah, so check out, I think it's, I'm trying to remember the name of the PI. I think it's Richard Sheltima, if I'm not wrong. I hope I'm not getting that wrong, but look on bioRxiv in the last week or two. There's some work now on PASEF for cross-linked peptides.
Okay, let's see. There are a few questions here of very technical nature. Let me just summarize. So a couple of questions relate to your separations, upfront fractionation, and, particularly on this run where you get 6,000 proteins in one run.
Yep.
You remind what the conditions were for that.
Right, so that was, I think, a 2-hour gradient with a sort of a 40 or 50 centimeter heated column with 1.9 micron beads. And that, so it's single shot analysis, but it is a sort of semi-long gradient. And the library in that case was not actually a very deep library. So that was all, the library was also from single shot analysis. And so I think for those particular patient samples, we actually could improve the data by doing perhaps a little bit deeper library, which we can do, right? And we can go back and analyze the same DIA data. That would be one option.
Okay, and again, compressing a couple of questions. You talk a little about the data analysis side. What are the sort of standard tools that people use, like Skyline and MaxQuant, practical for this sort of data? Is Spectronaut an option for this data as well?
Sure. So yeah, so we started with OpenSwath because this was a great place to kind of prototype things and get the method working. And this is still the tool that I'm using. In terms of analyzing the DIA PASEF data, Skyline is now pretty much fully compatible, I think. Spectronaut is also fully compatible, and I think people are starting to see nice data from using Spectronaut. I believe, so you mentioned MaxQuant. MaxQuant is compatible with the DDA PASEF data, but it's my understanding that they will have a DIA implementation. I'm not sure what the status of that is at the moment. And I think there are also some commercial, so Peaks, for example, I think can handle the DIA data. And DIA-NN, this neural net-based one, has a sort of an alpha implementation, I believe. So we're now coming to the point where, yeah, the data analysis is starting to kind of crank up and be usable. The data is bigger, so each one of these files is for a complex sample on the order of kind of 10 gigabytes, and the processing. So the processing does take a little bit longer than with standard DIA data, I would say, but I think the software developers are working on that.
Okay, that was exactly one of the questions was coming up was, can you comment on the amount of computational time to analyze the data in the timsTOF? I guess that probably doesn't have a single answer, but
it's a multiple of standard DIA data of something like I want to say something like 3 times or 5 times, something like that, 3 or 4 times. I'm not sure how precise that is, but it's certainly it's more. There's probably work to do there. Yeah.
Okay. Very simple one. When will DIA PASEF be available for commercial use? Do you know the answer to that?
Oh, good question. So I guess that's a question for Bruker now. I think some customers have this enabled in a sort of beta kind of way. And I know that the current software, acquisition software, is compatible with this, but you have to make this sort of not particularly beautiful looking table to implement it. And I guess it should be pretty close. So I don't know. I think I'm going to have to pass that question off to Bruker. I know some people have it. I don't know if it's completely generally available though, but it should be very close.
Okay. In the interest of time, I may want to move along to the next section. There are still a few questions here that we haven't gotten to, but they tend to be fairly technical ones. So perhaps you could jump in and answer those, or type answers to those. But in the meantime, I think we'll cut it off and move on if that's okay with you, Ben.
Sure. Thank you. Thanks very much.
Great.